Have you ever, I don't know, just sat there and thought about what makes us human? I mean, what really sets us apart?
Well, a lot of things, I suppose.
Yeah,
But
yeah, yeah, for sure. But there's this one thing, right? This kind of obvious one.
yeah,
Our brains. Like, they're so much bigger and more complex than any other animal, even our closest relatives. It's kind of crazy when you really think about it. And that's what we're diving into today on the deep dive. We're going to try and figure out how the human brain got to be so, well, impressive, right?
yeah, it's a question that's fascinated scientists forever, and there's some really cool new research that sheds light on it.
Okay, so tell me, what are we looking at today?
Well, we'll be focusing on a recent study from the German Primate Center and the Max Planck Institute.
Okay, so a pretty solid source right there.
Yeah, they're the experts. And we'll also be taking a deep dive into a super detailed research article that was published in Science Advances.
Cool, cool. What's it called?
It's got a bit of a mouthful of a title, but it's called, hang on, let me get it right. A Diet of Human-Specific NBPF14 and NOTCH2NLB orchestrates cortical progenitor abundance crucial for human neocortex expansion.
Wow, that is a mouthful.
Yeah.
So, a lot of big words in there, but I think I get the gist.
Yeah,
We're talking about specific genes that are, unique to humans, right? And they're involved in making our brains bigger.
exactly.
Awesome. So today's mission is to really get down to the nitty-gritty of how these genes work, to understand the mechanics of how they help build our bigger, more complex brains.
Exactly. We're going to pull out the key takeaways from these research papers, breaking down the science into something we can all understand.
Love it. Okay, so, just to set the stage here, the human brain is way bigger than a chimp's brain, right?
Oh, yeah, definitely. Especially if you look at the neocortex.
Hang on. Remind me, the neocortex, that's the part of the brain responsible for all the, higher level thinking, right?
Things like language, abstract thought, all that
Yeah.
good stuff.
Okay, cool. So how much bigger is our neocortex than a chimp's?
It's about 3 times larger. That's a huge difference.
3 times. Wow. And I think it's pretty clear that this size difference is a big part of why we can do all the things that chimps can't, right?
Mm.
Why we have language, why we can build complex societies, write symphonies, all of that.
Right. It's hard to argue against that connection.
Mm.
So the question is, how did our brains get so big?
Mm.
And the research points to a process called cortical neurogenesis.
Okay, break that down for me. What is cortical neurogenesis?
So basically, it's the process of creating new neurons, new brain cells, in the cortex during fetal development. It's like the brain's construction phase.
So we start with a bunch of starter cells and then they turn into all the different types of brain cells we need?
Kind of, yeah. And these starter cells, they're called cortical neural stem and progenitor cells, or CNPCs for short.
Okay, CNPCs. Got it. And these are the cells that are responsible for building our big, beautiful brains.
Yes, exactly.
So tell me more about these CNPCs. What are they like?
Well, there are 2 main types we're interested in. First, they're the amical progenitors, or APs.
APs, okay.
And these guys, they hang out in this area called the ventricular zone, or VZ,
Yeah.
which is like the inner lining of the developing ventricles in the brain.
Ventricular zone. Got it.
You could think of the APs as the initial pool of stem cells, kind of like the raw materials for building the brain.
Okay, so those are the APs, the initial pool. What's the other type?
The other main type is the basal progenitors, or BPs.
BPs, all right.
And these BPs, they live in a different zone, the subventricular zone, or SVZ, which is right next to the VZ.
So one zone's for the starter cells and the other zone's for the the next stage in the process.
Right, exactly. And actually, this SVZ is really interesting because in species with bigger brains, like humans, it's got these distinct inner and outer regions, the ISVZ and OSVZ.
ISVZ and OSVZ. Okay, got to keep track of all these acronyms. But why are these different regions important?
Well, that outer region, the OSVZ, it's particularly rich in a specific type of BP called basal radial glia, or BRG. And these BRG cells, they're strongly linked to the expansion of the neocortex. They're like those specialized builders for a bigger brain.
Okay, I'm seeing a pattern here. APs in the VZ, then BPs, including these special BRG cells. In the SVZ, it's like a little factory in there.
It is, kind of. So the typical way it works is that the APs, those initial stem cells, they produce the BPs. And then those BPs, they go on to make the neurons, the actual brain cells.
So it's like a multi-step process.
Exactly. Now, here's a really important point. Just having more APs, more of those initial stem cells, isn't enough to explain why our brains are so much bigger.
Really? Why not?
Because you also need a corresponding increase in the generation of BPs, those intermediate cells. Think of it this way. If you only have a ton of APs, but they're not efficiently turning to BPs, you won't end up with enough neurons to build a bigger brain. You need the whole production line to be ramped up.
Got it. It's about the entire process working in sync. Okay, so that brings us to the really interesting part. The genes. What is it about human genes that makes this whole process different?
Well, the researchers focused on genetic changes that are specific to human evolution. They were looking for those little tweaks in our DNA that might cause human CNPCs to behave differently compared to those in other primates.
So, like, are we talking big changes to our DNA or just tiny little adjustments?
It's actually a combination of both. There are small nucleotide changes, which are basically tiny alterations in the DNA sequence.
Okay.
But what's really fascinating in this case is the emergence of entirely new genes, genes that weren't present in our ancestors and evolved specifically in humans. These are called human-specific genes.
Whoa, human-specific genes, those sound important.
They are. And what the researchers were really interested in were the ones that are active in those CNPCs, the brain-building cells, rather than in the mature neurons.
So these genes are influencing the very cells that are constructing our brains, not just how the finished brain works.
Right. And actually, there's been previous research on this too, looking at other human-specific genes.
Oh, yeah, like what?
Well, for example, there's this gene called ARHGAP11B. It's been shown to increase the production of those BRG cells. Remember the ones in the outer SVZ?
Yeah, the specialized builders.
Right. And ARHGP11B also seems to make BPs renew themselves more.
So more specialized builders, and they keep making more of themselves. That sounds like a recipe for a bigger brain.
Exactly. There's also a family of genes called NOTCH2NL, and those have been linked to the size and behavior of those progenitor cell populations too.
Okay, so scientists have been on this trail for a while, looking at different genes that might explain our big brains.
They have. And that brings us to the two genes at the heart of this particular study, NBPF14 and NOTCH2NLB.
All right, NBPF14 and NOTCH2NLB. Those are the ones we're really focusing on today.
They are. And what's interesting is that they're located very close to each other on chromosome 1.
That's got to be more than just a coincidence, right?
That's what the researchers thought too. And they both show strong activity in those APs, especially the apical radial glia, or ARG, which are a subtype of APs.
So they're both active in those initial stem cells, the APs. Interesting.
Yeah, and NBPF14, that gene, it's part of a larger family of genes called the NBPF family.
Oh, a whole family of brain-building genes.
Well, we don't know that they all build brains, but they share this region called the Olduvai domain, or DUF1220.
Olduvai domain. Sounds kind of mysterious.
It is, kind of. We don't really know exactly what it does. It's a bit of a biological puzzle. But there's a hint from another member of the family, NBPF1. It seems like it might be involved in controlling energy within cells through something called mitochondria.
Mitochondria, the powerhouses of the cell. So maybe this whole NBPF family is involved in, like, powering up brain development.
That's the thinking.
Yeah.
And the researchers have this idea, a hypothesis, that NBPF14 and NOTCH2NLB, they might have actually evolved together as a kind of two-gene module.
A dynamic duo. I like it.
Yeah, and because they're both active in the same cells, those CMPCs, especially the ARG, the big question they wanted to answer was, do they work together in a coordinated way to affect how these cells behave?
Makes sense. It's like if they evolved together, maybe they do more together than they could do alone.
Exactly.
Okay, so how did they figure that out? Like, how do you even study something like that? You can't exactly go in and tinker with genes in a developing human brain, can you?
No, definitely not. They used some really clever methods. One of the things they did was use this technique called automated microinjection.
Sounds complicated.
It is, but basically they used it to deliver mRNA, the messenger molecule that carries the instructions from a gene, directly into individual APs in mouse embryos.
Why mice?
Well, mice don't naturally have these human-specific genes, so by putting them into mouse cells, they could see exactly what they did.
Ah, clever. So they could isolate the effects of just these genes.
Right, and they also used another model, chimpanzee cerebral organoids.
Whoa, chimp mini brains. That sounds wild.
Yeah, it's pretty cutting-edge stuff. They're not full brains, but they mimic key aspects of early brain development in chimps.
So a more realistic model for studying human brain evolution.
Exactly.
Okay, so they've got their mouse embryos and their chimp mini brains. What did they actually find? Let's start with NOTCH2NLB. What was it doing?
So when they expressed NOTCH2NLB, they saw that it led to an increase in the clone size of APs in the mice. Basically, one AP was making more copies of itself, or its immediate offspring.
So more of those initial stem cells.
Right. And in the chimp organoids, they saw a higher percentage of APs that were actively dividing and multiplying.
Makes sense. It's doing what we thought it might be doing, amplifying those APs.
Yeah, it seems to be playing a big role in expanding that initial pool of stem cells.
But like we said earlier, just having more APs isn't the whole story, right? You need those BPs too.
Exactly. And the researchers actually point out that NOTCH2NLB alone probably can't explain the massive expansion of the neocortex we see in humans.
Because you need that boost in BP production too.
Ex-
Okay, so that's NOTCH2NLB. What about its partner, NBPF14? What was it up to?
Interestingly, NBPF14 was doing something different. It didn't seem to affect how fast the APs multiplied, not in the mice or the chimp organoids.
So it wasn't making more APs.
No. But what it did do was increase the delamination of AP progeny from the VZ.
Okay, hold up. Delamination, what's that?
Think of it like this. Those daughter cells produced by the APs, they kind of lose their connection to the inner lining of the ventricle, the VZ, and they migrate out to the SVZ.
So they're moving from the starter zone to the next stage zone.
And in the SVZ, they have the potential to become those BPs, those important intermediate cells. And they saw this increase in outward migration in both the mice and the chimp organoids when NBPF14 was expressed.
So it's like NBPF14 is the one saying, hey, you guys move out of the nest, go become something else.
Yeah, that's a good way to put it.
And it's making sure there are enough of those BPs to build a bigger brain.
Exactly. And when they looked closely at the mice, they saw that the cells that delaminated when NBPF14 was active, they tended to take on the characteristics of BRG, those basal radioglia we talked about.
Right, the specialized builders in the outer SVZ, the ones that are really linked to neocortex expansion.
Yep, those ones. And in the chimp organoids, they saw an increase in a specific marker called HOPX, which is a sign of BRG cells, in the SVZ when NBPF14 was expressed.
So it's not just pushing cells out of the VZ, it's also encouraging them to become this specific type of BP that's important for a bigger brain.
Exactly.
Okay, this is all making sense now. NOTCH2NLB makes more APs, and then NBPF14 makes sure enough of them transition into BPs, especially BRG, to actually build a bigger brain.
It's like a perfectly coordinated system.
And did they, test what happens when both genes are active at the same time?
They did, yeah. And when they expressed both NBPF14 and NOTCH2NLB together, they saw this beautiful synergistic effect, meaning they worked together to make even more BPs. NBPF14 kept doing its thing, promoting that outward migration, even when NOTCH2NLB was around.
And what happened to the AP pool?
It either stayed stable in the mice or it didn't shrink in the chimp organoids. So you have this increase in BPs without losing those initial stem cells.
Whoa, that's amazing. It's like they both do their part, and it leads to a much larger pool of BPs to build a bigger brain.
Exactly. NOTCH2NLB boosts the initial supply of APs, and NBPF14 makes sure a lot of their offspring get converted into BPs. It's like increasing the production of raw materials and making the assembly line more efficient, all at the same time.
That's a great analogy. Now, I know the researchers also looked at how NBPF14 was actually doing its thing. They focused on something called cleavage plane orientation during AP division. Can you explain what that means?
Sure. When an AP divides, it can do it in different ways. If the division is vertical, along the main axis of the cell, it usually produces two new APs.
So more of the same.
Right. But if the division is at an angle, oblique or horizontal, then one or both of the daughter cells can detach from the VZ and move outwards.
To become BPs.
Exactly. And the researchers found that when NBPF14 was active, it increased the frequency of those angle divisions in the chimp organoids.
So that's how it's promoting delamination. It's influencing the way those APs divide.
Exactly. It's pushing them towards producing those BPs.
Yeah.
And NOTCH2NLB, it seems to favor the vertical divisions, which makes sense because it's boosting the AP pool.
And when they're both active, what happens?
Interestingly, the overall orientation of the divisions was more similar to the control, as if they were balancing each other out.
Wow. So evolution has given us this incredibly elegant system for controlling brain size. You have these two genes working together, one making more stem cells, the other making sure they transition into the right type of progenitor cell, and it all leads to a bigger brain.
Exactly. And the researchers talk about what might have happened if these genes hadn't evolved together. If NBPF14 had evolved alone without NOTCH2NLB, it might have actually been detrimental.
How so?
Well, all that delamination might have depleted the initial pool of APs too quickly, which could have limited the overall number of neurons produced, even with more BPs.
So it's like you need a balance. You need enough of both types of cells to really build a big brain.
Exactly. And if NOTCH2NLB had evolved alone, you might have ended up with a ton of APs, but not enough BPs to make use of them.
Another bottleneck. So it really is the combination of these two genes working in sync that's made all the difference.
That's the key takeaway. It's this finely tuned interplay that's allowed for the massive expansion of the neocortex in humans.
This is also fascinating, but I mean, does this have any implications for us today, like for understanding brain disorders or anything like that?
It might. It turns out that both NBPF14 and NOTCH2NLB are located in a region of chromosome 1 that's been linked to certain neurodevelopmental disorders.
Really, what kind of disorders?
Well, there's this one called 1Q21.1 distal duplication deletion syndrome. It can manifest in different ways, but it's been associated with autism, schizophrenia, and even differences in head size, sometimes leading to microcephaly or macrocephaly.
So a smaller or larger than average head, which would likely mean a smaller or larger brain.
Exactly. And the researchers suggest that NBPF14 might be involved in the brain size aspect of this syndrome.
Wow, that's a pretty big connection.
Yeah, it is. And they also pointed out that a previous study found that when NOTCH2NL genes were knocked out in organoids, it led to premature neuronal differentiation, meaning that the neurons were forming too early before enough progenitor cells had been made.
So again, it's all about that balance.
Right. They think that if NOTCH2NL isn't working properly, it could lead to an imbalance where NBPF14's effects aren't properly controlled.
That's really interesting, but also kind of scary, right? It shows how important it is for these genes to be working together properly.
It does. But of course, like any good scientific study, this one also raises more questions than it answers.
Well, that's how science works, right? Every answer leads to more questions. So what are some of the things that still need to be figured out?
One thing they pointed out was that there were some subtle differences between the results in the mice and the chimps.
Like what?
Well, for example, how NOTCH2NLB affects delamination. It didn't seem to have the same effect in both models.
So maybe there are subtle species-specific differences in how these cells behave.
That's what they think, yeah. And there are also some practical challenges in studying these genes.
What kind of challenges?
Well, both NBPF14 and NOTCH2NLB, they're very similar to other genes in their families.
So it's hard to target them specifically without affecting other genes.
Exactly. It's tough to develop tools that are precise enough to knock out or knock down their activity without affecting their close relatives.
Ah, that makes sense. It's like trying to untangle a really tight knot.
It is, kinda.
So where do we go from here? What are the next steps for this line of research?
They suggested a couple things. One is to use iPSC-derived cerebral organoids, which are mini-brains grown from human cells, from patients with that 1Q21.1 deletion syndrome.
So you can study how these genes function in a human context and specifically in people who have a disorder linked to them.
Exactly. And another big one is to really figure out the nitty-gritty of what NBPF14 does.
Like at a molecular level.
Yeah, to identify all the proteins it interacts with. That'll get us a much better understanding of its role in this whole process.
Awesome. It's like the more we learn, the more we realize how much we don't know.
It is, definitely. But it's exciting, right?
Oh, absolutely. It's like peeling back the layers of this incredible mystery that is the human brain.
And this study, it's really important because it's one of the first to show how two human-specific genes work together to control brain development.
Yeah, that synergistic effect. They're more than the sum of their parts.
Exactly.
So, like, for everyone listening, what's the big takeaway message here? What have we learned today about how our brains evolved to be so large and complex?
I think the biggest takeaway is that it wasn't just one thing. It wasn't like one gene suddenly mutated and boom, we had bigger brains. It was a series of changes, a whole bunch of genes working together in a coordinated way.
Yeah, like NBPF14 and NOTCH2NLB. They're a perfect example of how evolution can fine-tune these intricate genetic interactions to achieve something truly remarkable.
Exactly.
And this isn't just about understanding our past, right?
No, not at all. This kind of research can help us understand how the brain develops in general, and that can have implications for all sorts of things, like treating brain disorders and developmental problems.
Absolutely. And it leaves us with some pretty mind-blowing questions to think about, like what other genetic partnerships might be out there shaping our brains and making us who we are?
Yeah, and what happens when those partnerships go wrong? It's like we've just scratched the surface of this incredibly complex and fascinating system.
Totally. It's enough to make your head spin, but in a good way.
Yeah, a good way.
Thanks so much for taking us on this deep dive. It's been an amazing journey.
You're welcome.
And for all of you listening out there, keep those brain cells firing, keep those questions coming, And we'll see you next time on The Deep Dive.